So today we're going to talk about facilitated diffusion, which is a passive mechanism of diffusion. And so being passive, all that means is that it's moving according to the concentration gradient. All right, but facilitated, the term facilitated indicates to us that it's not going to be just simply moving across the membrane. It's going to need some help. It's going to need to be mediated or regulated. And so we have transmembrane proteins that are embedded into the membrane. And we talked about these transmembrane proteins. If anyone remembers from that phospholipid image that we drew, we talked about the different features of the phospholipid bilayer. And so transmembrane proteins were those integral proteins we talked about, the ones that are permanently fixed into the membrane. And so these proteins will not leave the membrane unless this cell is damaged or destroyed, right? They're permanently embedded into the membrane. Now they're highly specific, meaning just like enzymes, they're only gonna bind one type of molecule or one type of particle at a time. They're also one-sided, meaning that because they have to undergo this conformational shape, what we'll see is that the protein interacts with a molecule on one side of the membrane, has a shape-shifting conformation, and then the molecule can interact with the other side of the membrane as well. So let's look at what that could look like. So we have our plasma membrane, our phospholipid bilayer, the extracellular space, the intracellular space. This is an example of our transmembrane glucose carrier or glucose transporter. And so as the name implies, glucose cannot simply move through the membrane, right? It needs to be facilitated by one of these transporters. And so when we say that it's one-sided, we mean that glucose can interact with this side of this transporter. The transporter will sort of enclose and accept glucose. There'll be a shape-shifting or conformational change such that glucose will be exposed to the internal surface of this cell. And in such a way, this transporter is facilitating the movement of glucose through the membrane, or facilitated diffusion. Right? Important to remember here is that this is still according to the concentration gradient. So there's still more glucose on the extracellular environment outside the cell than there is inside the cell. So the driving force for this is not different than simple diffusion. They're all passive. They're working according to a gradient. With active transport, we'll see that that will differ some. Okay, so this is facilitated diffusion. Let's look at some of the factors that affect how fast this can go. The first one is going to be the weight of each carrier, which only makes sense, right? Each specific carrier, there are tons of different transporters in the body, and they all have an innate ability or innate number of molecules that they can transport per second. We're going to see for glucose, it's about 10,000 glucose molecules per second. Okay, and so that will affect how fast glucose can be transported. The number of carriers in the membrane, this is really important, and this ties back to homeostasis. If we need to, if the body so needs to, we can upregulate the number of carriers on the membrane. So we can embed more GLUT transporters in the membrane. That will facilitate the faster movement of glucose into the cell. And again, this will depend on your physiological state, whether you're in the fed state or the fasted state, et cetera, et cetera. And the opposite can be true too. We can remove those GLUT transporters from the membrane. We can downregulate their function in the membrane if that's needed. The concentration gradient, right? So as we just mentioned, because this is passive transport, although it needs to be facilitated, it still is happening according to the concentration gradient. That's what makes it passive, right? That downhill direction, that spontaneous nature of this type of transport, okay? Now, when we look at this graph, we're plotting the net flux, and just to remind ourselves, this is the net accumulation of particles, or the net movement, against the concentration gradient. So what the concentrations of either side of the cell look like, and whether or not there's a gradient. And what we'll see is that these two properties show a directly proportional relationship until they get to this point where they just level off. Right, now this looks very different from this graph, right? Or this graph. Here we saw that there was an indefinite directly proportional relationship. And this was simple diffusion, not being facilitated, just moving right through the membrane, no transporters, no carriers, no channels. Whereas when we use a transporter, we'll see that this graph starts to level off. Can anyone explain why or think of why we don't have that indefinitely direct proportional relationship? Not so much, so not equilibrium, but something else physiological happens. Yes, that's correct. So the transporters sort of get saturated. They can only move a certain amount of molecules across the membrane at a time. Okay, whereas with simple diffusion, did we have anything to be limiting? Did we have a transporter or a carrier or a channel? We didn't. So that graph would be directly proportional, it would be indefinite, whereas this would sort of level off because this is the point where our transporters are maxed out, they're saturated. Okay, so this will not be indefinitely or show an indefinite line the way that the other graph did, okay? Let's look at an example of facilitated diffusion, looking at one of the GLUT4 transporters. Now again, we mentioned the function of this transport is to move glucose into the cell. This is how we can absorb glucose, okay, and be able to use it inside the cell for energy. These proteins are synthesized on the rough endoplasmic reticulum, so right away that tells us that they're made for either membrane structure or export, right? That should be something that we know at this point. Where a protein is made dictates whether it's going to be used internally in the cell or be exported or used in the membrane. So it's made in a rough ER, it's going to be exported or embedded in the membrane. So it's packaged in vesicles, transported, inserted into the membrane, and this is how we facilitate the movement of glucose into the cells. Now in the case of diabetes mellitus, we don't have insulin acting as it should, and so we have less of these GLUT4 transporters in the membrane. And as a result, we cannot uptake glucose as efficiently. And so what do you think happens? If we can't bring glucose into the cell, where does it linger? In the bloodstream, right? In the bloodstream, perfect. So because it's lingering in the bloodstream, this is where we start to get our pathology, this is where we start to get our complications. Glucose is quite injurious, it's damaging to blood vessels, small blood vessels of the eyes, of the feet, of the fingers, of the kidneys, right? And so this is what becomes problematic, these GLUT4 transporters. We can uptake less glucose, and so there's more glucose lingering in the blood. In the case of diabetes type 1, which is where we have insulin insensitivity, a similar thing happens, but the only thing is we have insulin, our cells are just not receptive to that insulin, right? Okay. All right, so that was facilitated diffusion that required a transmembrane protein, which was a carrier or sometimes called a transporter. Okay, important to distinguish a transporter or carrier from a channel. They look very different structurally, and they have some different properties which we'll talk about. So we also want to remind ourselves here, although this is using a channel, this is still passive transport. So this is still taking place according to that concentration gradient that we talked about. So some features here, this requires a protein. So this is also requiring a transmembrane protein, but this is not a carrier or a transporter, so make that distinction. The structure of this protein is such that it looks like a passageway or a pore, hence the term channel, right? This is still not simple diffusion, still passive diffusion, right? There are three types of passive diffusion: simple diffusion, facilitated diffusion, channel mediated diffusion, okay? I hope that's clear. So not simple because we're using some sort of mechanism or some sort of transporter, but it still is passive because we don't require energy and because we're using that concentration gradient. So this protein, the structural protein, is going to function like a passageway or a pore, and it's going to allow the movement of whatever molecule you're trying to get through the membrane. It's going to go right through that passageway or right through this pore. And so what we see are these polypeptide subunits. That just tells us that this is a protein structurally. And we talked about protein structure, right? That quaternary or tertiary structure forms itself in such a way that it looks like this channel and sits in the membrane. And again, this is permanently embedded in the membrane because it is an integral transmembrane protein. Now, these are also going to be substance specific. meaning that only certain molecules can go through these. We'll talk about the different types of channels, but also substance specific, just like we saw with transporters. Now, we want to point out here that whereas with facilitated diffusion, which required transporters, we saw that there was one-sided movement, right? So the molecule in question interact with the protein on one side, there was a conformational change, it was released on the other side, et cetera, et cetera. So that happened on one side at a time. With channels, we'll see that there's bidirectional movement, meaning that a molecule, whether it's water, we'll talk about water moving through some of these channels, whether it's an ion, we'll talk about what the different molecules that go through, but whatever it is, it can go through in both directions at a time, right? Not that, that transformational shape that we saw with the transporters. All right, so these are some of the flavors or types of channels that we have. We have aquaporins that exclusively allow water. So the movement of water molecules happens through aquaporins, and so as a result, we'll see aquaporins in the large intestines, right? These are areas where we have lots of water reabsorption, the collecting duct of the kidneys. And when we want to reabsorb more water, we simply embed or upregulate the number of these channels in the membrane, okay? Then there are ion channels that allow the movement of ions, right? We want to remind ourselves ions are charged particles. They're not going to move freely through that lipid bilayer. And so they require some sort of mechanism to facilitate them or to allow them to pass. There are leak channels that allow the free movement of ions. We'll talk about some important leak channels which help to maintain the sodium, potassium, ATPase together with that pump. They maintain the relative ion concentrations inside and outside of the cell. Gated channels are also very important. We're going to look at those a lot throughout the semester, especially in muscle and in the autonomic nervous system. And then you also have bidirectional ion channels, which just allow the bidirectional movement of some ions. Okay? Leak channels usually allow the one-way movement of some ions. Factors that affect the rate of transport of ions are going to be very similar to factors that affect the rate of transport to our transporters, right? The individual or the innate ability of each of the channels. Okay, we said that that could differ by tissue type, by location, and so each channel has an amount of molecules that it allows to pass through each second, and that can change. The number of channels that are in the membrane can also change. As we said before, you can upregulate to embed more, you can downregulate to embed less. If you're in the desert and you lack water, you want to reabsorb as much water from your kidneys as possible. So you're going to upregulate the number of aquaporins in the collecting duct. That's going to help you reabsorb more water. If you're well hydrated, you don't need that mechanism as much. You're going to down-regulate the number of aquaporins, and that's going to decrease the amount of water that's reabsorbed in your kidneys. And so we can change the number of channels that are in the membrane itself. And then the open or closed state of the membrane, of the channel, can dictate the rate of movement as well, whether that channel is in an open state or a closed configuration.